good morning, students. Today we are going to start the new genus, that is Chara. But before we start this new genus, let's have a quick revision of family Charophyceae. Freshwater, we have already discussed it. Multilayered structure may be present. It's not compulsory that all the genus will have multilayered structure, but yes, multilayered structure may present. Motile cells asymmetric and scale common outside of the motile cell. मोटाइल सेल वो कुछ भी हो सकते हैं, जूस्पोर हो सकती है और नियोजूस्पोर हो सकती है. And the motile cell two laterally or subapically inserted flagella. Unlike Chlorophyceae where both the flagella are terminally placed, here you will see that the flagella may present laterally, means or subapically. Rhizoplasts not present, and instead of that you will find microtubular roots. Isport not present, and phragmoplast produce new cross walls after cell division. That we have already discussed, that sexual reproduction results into dormant zygote, and meiosis will occur in that zygote at the time of germination. And unicellular alga mesostigma can say that is the closest extent relative to the ancestral green flagellates. अभी हम बात करें all these features, they are slightly different than the green algae and they are slightly common with the bryophytes. But this is the genus which shows more feature of green algae and less feature of Charophyceae. Glycolate broken down by glycolate oxidase and urea broken down by urease. So these were the some features. And you already knew that Fritsch classified this class in 4 orders. Order number 1, Clepsomydiales. These are the simplest Charophyceae which are unbranched filament without holdfast and plasmodesmata absent. Zoospore here you will see neck. Whereas Zygnema you can see that sexual reproduction takes place by conjugation. And I am again saying, when we are saying that sexual reproduction takes place by conjugation, don't never get confused with the conjugation of the bacteria. It's altogether different. Coleucetase we have already discussed, where we have seen the heterotychous condition. Okay, Setae with present. So all those features we have already discussed in the previous class. Today we will discuss the one of the most advanced order, that is Charales. Oogamous sexual reproduction, sterile cells around antheridia and oogonia. यहाँ पर आप जो देखोगे antheridia and oogonia के बाहर sterile cells मिलेंगे आपको, and that make it different than the typical green algae or normal algae, because in algae we don't assume that our reproductive organs will enclosed by sterile cells, but here it's occurred. Okay. And surprisingly, there is no zoospore take place. There are very few order, irrespective of different classes of algae, where zoospores not take place. But yes, here zoospore don't take place. Its body is comparatively the most complex in green algae. Okay, the more complex body you will see only in brown algae or some red algae. But few genus which have this type of complex plant body, because it is divided in nodes and internodes. So let's start discussing the individual Chara, which is the member of Charophyceae, and that Charophyceae is the only family of this order, Charales. Before we start, there was some confusion regarding the placement of Chara, like pigment, green pigment, chlorophyll A and B, only Chlorophyceae. According to Fritsch, only Chlorophyceae and Eugleninophyceae. There are only 2 classes which are having chlorophyll A and B. Rest of the class having chlorophyll A and some other pigment, but there is no other class which is having B. That's why fridge kept or colloquials in green algae. Reserve food material is starch. That's why we will give also use staining material while making the temporary slides. Iodine. Haploid nature of the plant body, just like Chlamydomonas, Volvox, Oedogonium. We have seen that the colloquid as well as Chara will have the most dominant phase in their life cycle, that is haploid. But apart from these common sharing of the feature with the green algae, they have some distinct features as well. Erect vegetative plant body with distinct nodes and internodes. Okay, you haven't seen till date which is having a plant body or the algal thallus divided into nodes and internodes. But here, yes, you will see it. Dimorphic world of lateral branches and the occurrence of the sex organ only in lateral branches. So they resemble with the higher plant because of these features. And that's why some scientists kept them along with other embryophytes in a different group, that is streptophytes. That many carriers calcified with the concentration of plant on the bottom of lake, leading to the formation of malt. Malt क्या होता है आपका? Calcium carbonate and magnesium carbonate deposition. And that's why these members are also known as stoneworts. So someone is asking, give an example of stonewort, you can say Chara, or you can, Chara is also known as stoneworts. Is it clear?
Yes, sir.
Yes, sir.
You knew that any plant or any animal system which is having this kind of deposition will be the best candidate to make its fossil. This calcified had been termed as gyrogonite. Gyrogonite. Compared to any other algae, you will see more fossil of this group. अब बात करते हैं Chara की habit, as we already knew that it's a freshwater macroscopic. Macroscopic का मतलब
Visible with eyes, naked eyes.
Yeah, we can see it with our naked eyes. Filamentous and multicellular. इसके बाद आते हैं its habitat, its epipellic. What do you mean by this epipellic? What do you mean by pellic? Pellic means mud. Epi means bahar. Means when a plant is growing in a mud, or sorry, on a mud, not in a mud, that is known as epipellic. If it is endopellic, it's meant inside mud. कीचड़ के अंदर होगा तो एंडोपैलिक, कीचड़ के बाहर होगा तो एपिपैलिक. ग्रेजेरियस एल्गा, ये टर्म शायद आप लोगों ने पहली बार सुनी होगी, या हो सकता है ये बहुत ज्यादातर लोगों ने पहली बार सुनी हो. द मीनिंग ऑफ दिस, wherever you will see this alga, you will find it in groups. They are attached with the help of roots, like rhizoids, not that true roots, क्योंकि अगर ये true root होंगी then it will not be an algae. And again, stagnant, shallow, slow running freshwater bodies like pond, pools, lakes and stream. But एक ही condition है, either ये तो standing water हो or very slow running water. Whenever you are going to search for the Chara, try to locate that water body which is either having the stagnant water, or if it is a running water, the flow rate should be very slow. अब हम बात करते हैं इसके थैलस के स्ट्रक्चर की. The number one condition, plant body is complex macroscopic. जैसा आपको दिख रहा है ऐसे दिखेगा. I will show you the actual fixed material as well. Okay, but it's a diagrammatic representation of Chara. You can see the bottom or the underground or under mud portion is rhizoids, and the normally it can reach up to 20 to 30 centimeter. That's why it is known as macroscopic. The main axis is differentiated into nodes and internodes, and the internodes is a long, cylindrical, undivided cell inseated by a layer of corticals. This is node, this portion is node, and this portion is node. And in the area between these two nodes is known as internodes. यहाँ पे एक नोड है, यहाँ पे एक नोड है, जैसे गन्ने होता है ना, गन्ने में दो गांठें होती हैं. The same way you will see one node over here, another node over here, and in between you will find internode. The rhizoids have oblique septa. It's a characteristic feature. There is one more term, statoliths. It's the structure, statoliths. What does it mean? That they are the specialized amyloplasts involved in the perception of gravity. Before going to that next slide, you should know the main function of rhizoids. जो roots के function होते हैं plant में, the same way you can imagine the function of rhizoids for Chara, fixing the plant, absorption of minerals, and reproduction and the pollination by bulbil and secondary protonema. From each node, this was the node, this was the node, this is node, this is a node, this is a node, this is a node. We are talking about only the main axis. If we focus on the main axis, we will find 4 different kind of appendages. Number 1, axillary branch or branch of unlimited growth. आप ये जो ब्रांच देख रहे हैं, is it visible to everyone? जो लंबी लंबी ब्रांचें निकल रही है इससे, can you see it?
Yes, sir.
Yes, sir.
This one, this, and this. These are known as branch of unlimited growth. Means there is no limit. They bear a resemblance to the main axis. जो feature main axis के होते हैं, the same feature you will see in the branch of unlimited growth. Means they are divided into nodes and internodes, and their internodes cellular arrangement is almost same like your main axis. जरूरी नहीं कि ये हर किसी निकले. You can see from this node there is a origin of branch of unlimited growth. From this node there is one more branch of unlimited growth. From this node one more is there, but there is no branch of unlimited growth from this node or this node. It's not necessary that each and every node will produce a branch of unlimited growth, but yes, there are some nodes which can give rise to branch of unlimited growth. They're differentiated into nodes and internodes just like main axis. जैसे main axis को आप बोलते हैं ना, यहाँ पे node और internode है. Same way you can see that this is node, and in between it is internode. This is node, and in between area it is internode. So they are exactly same like the main axis. This is the main axis. इसको आप label please कर लीजिएगा. And they have the primary laterals inward, and axillary branching arise from the primary internodal cell situated below the basal node of the oldest branchlet. So these 2 points we will discuss in the next slide. तो अगर हम बात करते हैं कि unlimited growth की, they can arise from any node but not from all nodes of main axis. They are having exactly the same like your main axis because they are also divided into nodes and internodes. What are those primary letters? From each node apart from branch of unlimited growth, you will see the branch of limited growth, which are known as branchlet or primary laterals. These are the primary laterals जो वर्ड्स में आपके निकलते हैं. These are short branch which arise in world. Means world means एक गुच्छे में निकलेंगे. You can see here they are arising in the world. चारों तरफ से निकल रहे हैं. Main axis. This is the main axis. It may be anything. This portion may be anything. It may be your main axis or it may be branch of unlimited growth. But from this node you can see there are 1, 2, 3, 4, 5, 6. 6 vacancy. हो सकता है इससे भी ज्यादा हो. These are the primary laterals or the branch of limited growth, and they always grow in world. Means They are characterized that I already told you. They are the limited growth because they have a limited number of the nodes and internodes, unlike your main axis as well as branch of unlimited growth, क्योंकि उनकी कोई लाइन की कोई लिमिट नहीं होती, whereas you will find a definite number of nodes and internodes in branch of limited growth. आप इसको भी अगर ध्यान से देखेंगे, this is node, this is node, this is node,
Yes,
this
sir.
is node, this is one node. You can enlarge this portion, this one. This is one node, this is another node, this is one more node. So it is also divided into nodes and internode, but unlike branch of unlimited growth and main axis, their number is definite. That's why they are known as branch of limited growth. The primary laterals, you will see some unicellular spine-like secondary laterals. From the primary lateral, you will see some spine-like structure. अभी इस पे ध्यान नहीं देना है, जो ऑरेंज और रेड कलर का आपको या ब्राउन कलर का आपको जो स्ट्रक्चर दिख रही है, don't focus on that, just focus on these spine like or lateral branching like things. Okay, they are not divided into nodes and internodes, so they are known as secondary laterals. And on the node of primary lateral, they are also having sex organ. The branchlet develops, ये point पहले आना चाहिए था, from the peripheral cells of the nodes. Third appendage, that is stiploids. Two stiploids develop at the base of each branchlet in most of the species. These are the unicellular outgrowth which arise from the basal node of each branchlet or primary laterals. वो हम जो पहले ही बात कर चुके हैं secondary laterals की, इसी के बारे में. These are the third one. The diagrammatic representation. The thick one is the primary laterals, okay, on which you will see the sex organ, and here you will see the secondary laterals or stiploids. This is node, this is node, this is node, and its internode. If you will cut it, you will find this kind of a structure. There is a central cell, and it is surrounded by the cortical cells or the peripheral cell. और जो हम बात कर रहे हैं ना, all the branching, whether it's primary or secondary or sex organ, they arise from the peripheral part only. कुछ भी डेवलपमेंट होती है, कह रहा है कि कहीं सी भी पार्ट से, इट्स स्टार्ट फ्रॉम द पेरिफेरल और कॉर्टिकल सेल्स, नॉट फ्रॉम द सेंट्रल सेल. कॉर्टिकल सेल्स के बारे में हम डिस्कस हम पहले ही कर चुके हैं. दे आर वर्टिकली इलोंगेटेड रोज, ये लंबी लंबी एक एक सेल्स होती हैं. So you can see there are total 4. One is the auxiliary branch or the branch of unlimited growth. The second was the branchlet or the primary laterals, and the third is the secondary laterals, and the fourth is the cortical cells. So this is all about the morphology of this. So this is a video. You can see it's, we can see it without using even a hand lens. Okay. Generally it's green in color because we have already fixed it, it has lost its pigmentation. That's why it's become white. And instead of visualizing it under the compound microscope, we will visualize it under a dissection microscope. अगर कोई बड़ी चीज है उसको डिटेल पढ़ना है, तो you should always use a dissection microscope or a hand lens only. So this is the view under the dissection microscope. Are not visible. आपको दिख रहा है ये? These are the sex organs.
Yes,
sex
sir.
organ हम बाद में बात करेंगे. But you can see the nodes, internodes. Okay, the word of the branches or branch of limited growth, and आपको nodes and नोड बिल्कुल क्लियर दिख रहे हैं यहाँ पे, एंड सेकेंडरी लेटर्स पे आपको दिख रहे होंगे बीच बीच में। ये देखिए स्पाइन लाइक स्ट्रक्चर, यस और नो?
Yes, sir.
Let's try one more
Yes, sir.
time. It's very difficult to hold a camera. Yeah, this is one more structure. So when you have this kind of material, try to take out these branches while observing it under the dissection microscope. And after taking out the lateral branch, now we will see how we will prepare the slides. This is the small part of the lateral branch. हो सकता है आपको भी ना दिखाई दे या दिखाई दे. You can enlarge it at your end. You can see there is a thread-like structure. Is it visible to everyone?
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay. It will be more clear once we will stain it. We are giving it to stain of acetocarmine, but why not from iodine? सोचेगा इसको मैंने acetocarmine से क्या है? So why acetocarmine? Why not iodine? Take one more slides. Take a small amount of glycerine. With the help of a brush, transfer it very gently. While using a needle, you should take extra care, because now you can see those dot structure on these threads. Can you see these?
Yes, sir.
दिख रही है dot structure? These are the sex organ of Chara. दिख रहा है आपको यहाँ पे क्लियर?
Yes, sir. Yes, sir.
After that, you will take a cover slip and cover it. Because it's a thick material, so you will need more glycerine. If it is a thin material, you will require less glycerine. उसको पूरा cover करने के लिए. And the method will remain same. Hold it in 45 degree and gently place it over the material. Because it's a thick material, your cover slip will not be placed properly. You can see one corner, it's not properly placed. That's why you have to gently press it. Is it clear कि आपको थोड़ा सा gently press करना है, ज्यादा जोर से नहीं करना, इससे glycerin सबको cover कर ले. And then you will observe it under the microscope. I will say one more thing. We show you only the preparation of one slide, that is stained with the acetocarmine, but we have also used slide preparation with stain of iodine. हमने iodine का आपको आंसर पता है, because it contains starch, that's why we are using iodine. But why acetocarmine? Just think over it. Let's discuss something about reproduction. As we already said that asexual mode means zoospore, aplanospore, hypanospore, nothing is there in Chara. It either reproduced by vegetative mode or sexual mode. Please remember this point. Vegetative reproduction, bulbils, amylin star, and the secondary protonema. Bulbils, these are the tuberous structure formed either on the rhizoids or on node of the main axis. This is the bulbil. Rhizoids पे मिल गया oblique septa के साथ. और you can see the rhizoid, this is amorphous. हो सकता है कि एक single structure की तरह आए, और it may arise in the multiple structure. Like in our case, in our material we have seen. Here you will see that this photograph is taken from the material which has been stained with iodine. So these are those bulbils. तो आपको यहाँ पे दिखा दिया हमने photographs जो rhizoids पे bulbils होते हैं, and here we can see the bulbils which are on the stem. Amylum stars, in some cases an aggregate of cells looking like a star. It develop on the lower node of the main axis. They are full of stars, means full of nutrition, and once there is a some stress condition is there, so that at that time these star-shaped structure can remain in dormant state, and when the environment is suitable for them, they will start germinating again.
Sir.
I hope you have seen those outgrowth on the nodes of sugarcane as well. Have you seen
Yes,
that?
sir.
Yes, sir.
Yes, sir.
Just see the beauty of nature. We are talking about algae, and I'm resembling it with one of the most advanced plant, that is sugarcane. Monocot की हम बात कर रहे हैं. We cut those sugarcane in small part, and we take out that part which having that outgrowth, because it is full of nutrition. The same way, these star-shaped outgrowth or these bulbils like outgrowth can help this Chara to survive in the extreme condition, and they will help it in the germination for the next season.
सर ये
There is no
आउटग्रोथ हुई सर, जहाँ पे ये सर जो नोड्स है ना, यहीं से सर एक न्यू प्लांट जर्मिनेट हो जाता है.
बिल्कुल बिल्कुल.
Yes.
सेम फॉर सुगरकेन, सेम फॉर कैरा. इस तरह के कुछ सेकेंडरी प्रोटोनेमा भी निकल सकते हैं. कैरा आल्सो रिप्रोड्यूस बाय प्रोटीनिमा लाइक आउटग्रोथ प्रोड्यूस. उसकी फोटोग्राफ मेरे पास नहीं है. From primary rhizoids or from the nodes of the old plant. But every time, whenever it is a growth, it will arise from the node only. चाहे वो amylum stars हो, bulbils हो, और secondary protonema हो. अब वो bulbil भी चाहे वो amorphous हो या normal bulb-like structure हो. Okay, this photograph is the stem bulbils, single structure, and this is the original photograph from our own sample जो हमारे labs में है. इसमें we will find the amorphous bulbil. Now let's discuss with the most fascinating things in the Chara, that is sexual reproduction. Before we start sexual reproduction, I'm asking you one more time. किसी को आंसर मिला कि एस्टोकार्मिन क्यों यूज करते हैं? सेकंड क्वेश्चन ये है, why we are using एस्टोकार्मिन?
सर एसिटोकार्मिन क्रोमोसोम से सिंपली बाइंड होकर सब कलर प्रोवाइड करता है.
तो why we are using it over here? और यहाँ पे कर रहे हैं तो बाकी जगह क्यों नहीं किया? चलिए सोचिए इसके बारे में. We will discuss it. Sexual reproduction is oogamous, and it is extremely advanced oogamous type. You will never see such kind of advanced reproduction in any other algae. The sex organs are so big that you can see it by your neck. Right? आपने देखा होगा कि when we have taken out the branch of limited growth on the slide, the dot like structure, although not that much detail, but yes, we can see that there is something on the male sex organ is a spherical, bright yellow or red structure, commonly called as antheridium or globule. ये जो structure है, it's either red structure or bright yellow structure. Whereas female sex organ is an oval, green or yellowish green structure, which is known as nucleus. This structure is known as globule. This structure is known as nucleus. Most of the species are homothallic or monoecious, but few are heterothallic or dioecious. Okay, what do you mean by homothallic?
So homothallic means that both are on one only. The both male and female are on one branch, or you can say
Both
branch.
male and female sex organ are on single thallic.
Yeah.
That is known as homothallic or monoecious. And if they are originating on different thallic, they are known as heterothallic. Good. Homothallic species are protandrous. I have already used this term. बताइए.
The male germinate before, develop before the
Yes.
female.
बिल्कुल बिल्कुल सही. What's your name, dear? Ananya? No.
Yes, sir.
If male reproductive structure is originating before female in a homothallic structure, homothallic thallus, that it is known as protandrous, and the nucleus is supported by pedicel, and the center is the oogonium with its single egg. I think this is the biggest egg cell of algae, or the algae we have studied till now. And the 5 spirally twisted tube cell over the oogonium, except at the tip where there are 5 corona cells. Corona देखो यहाँ पर भी आ गया. These 5 cells, twisted tube cells बोलते हैं इनको, cover this egg cell, and these egg cells are sterile in nature. Just take 5 threads like this, means parallel to each other, and try to wrap anything with the help of those threads. In the same manner, this egg cell get wrapped by these 5 twisted tube cells. From top to bottom, there are only 5 cells. So imagine करना पड़ेगा, like this is the 1, 2, 3, 4, 5. These are the 5 cells. Then they will come back after a rotation. Then they will 1, 2, 3, 4, 5. Means it is the same 5 cells which are wrapping this entire structure. And on the top, they are free, which makes the corona. कोरोना का मतलब जानते हो ना क्या होता है? Have you ever heard that term corona?
Maybe crown.
Yes, crown. Same way, the corona तो अब सब लोगों ने सुना ही होगा. कोरोना डिसीज सुनी है ना सबने? कोरोना वायरस.
Yes, sir.
यस
ऐसा
सर.
कोई है जिसने ना सुना हो?
एक्सपीरियंस भी की होगी सर. कई लोगों ने हो सकता है एक्सपीरियंस भी किया हो.
Yes, sir.
ठीक है,
Yes,
चलिए
sir.
मैं ये दुआ करते हैं कि जल्दी से जल्दी ये कोरोना चला जाए. हम केवल कोरोना इस न्यूक्लियल के ऊपर के स्ट्रक्चर को पढ़े, ना कि कोरोना डिसीज को. And when the nucleus is mature, the spirally twisted tube cells separate from one another cell just below the corona. यहाँ से फिर लूज होना स्टार्ट कर देते हैं जब ये मैच्योर हो जाता है. So this was all about the structure of nucleus. Let's discuss something about globule. The globule or antheridium occur below the nucleus. आप प्लीज का ध्यान रखिए, हो सकता है कि फोटोग्राफ लेते टाइम तो आपने ऊपर नीचे लिख दिया हो, बट द एक्चुअल ओरिएंटेशन इज दिस ओनली. मींस अपर साइड इज द न्यूक्लियस, lower side is the globules. And when you are showing it in the cell or under the microscope, make sure that the orientation will be like this only. The median cell develop into manubrium, and the 8 inner cell are transformed into primary capitular cell, which give rise to the antheridial filament. No need to go in that much of detail. ये जो main cell है, it is the main manubrium, and which give rise to the antheridial filament. आपको ये filaments इस तरह से दिखाई देंगे. यहाँ पर जो दिख रहे हैं ना, I will show you in the actual photograph. You can see thread-like structure inside this. These are the antheridial filament, and again they are remain inside a sealed cell. जैसे इसमें एग सेल में एक ट्यूब सेल से कवर होता है, ये सील्ड सेल से कवर होता है. जो ये इंटरडियल फिलामेंट, इट इज मेड मल्टीसेलुलर फिलामेंटल स्ट्रक्चर, and each of these cells can give rise to the single anthrozoids, which is having 2 flagella. When the anthrozoids are mature, the sealed cell of the globule separate from one another. In the antheridial filament cell will get break down, and it will give rise to the free anthrozoids. Iodine stain, though reproductive structure with the iodine stain. You can see what you can see only the female body. Yes or no? मेल
Yes,
स्ट्रक्चर आपको दिखाई दे रहा है?
yes, sir.
मेल
No,
आपको
sir.
मेल स्ट्रक्चर दिखाई दे रही है? मेल स्ट्रक्चर इसमें नहीं दिखाई दे रही है. को 2 रीजन हो सकते हैं. either while making the slide it has been broken down, और देखो कितना अच्छे से नोट दिख रही है यहाँ पे. just have a look. Corona nodes, those bulbils, egg cell, spirally twisted cells, everything is clear over here. एक बार और देखते हैं कोशिश करके. And you can see the cortical cell surrounded the main cell, center cell. Here, okay, यहाँ पे आपको जरूर दिख रहा होगा ये male structure. So this was the stain of iodine. Now I will show you the still photograph. These are the still photograph of when we stain it with the iodine. Here you can also see the male structure. Clearly visible है ये. Female and male both are clearly visible.
Yes, sir.
Okay, सिद्धार्थ, आपने बोला कि आपको मेल और फीमेल इक्वली विजिबल है. चलिए नेक्स्ट देखते हैं अब. दिस इज वन मोर स्लाइड जिसमें हमने क्या किया, वी ट्राई टू ब्रेक इट. इसको जैसे पंच करेंगे ना तो सील्ड सेल, ये जो सील जो कवरिंग है इसकी, इट विल गेट ब्रेक डाउन एंड इट विल रिलीज दिस एंथ्रीडियल फिलामेंट्स और ये बाहर निकल के आ जाएंगे. कुछ और ज्यादा कुछ नहीं किया हमने. सेम आयोडीन स्टेन है. अब हम करते हैं एसिटोकार्मिन स्टेन. सिद्धार्थ आप ही ध्यान से देखिएगा, अब स्पेशली आपसे. Is it properly visible now?
Yes, sir.
Are they equally visible now?
The antheridial filaments are visible now.
And in the previous slide,
They were not visible.
they were not visible. Okay, this is one more slide. Okay, although there were some air bubble were also there, but we have used this. You can see antheridial filament is there. Okay, this sealed cell is still not visible. Sealed cell देख रहे हो ना आप इसका? ये अभी भी visible नहीं. These sealed cell are still not visible. But we were trying to focus it more on the antheridial filament. It's very difficult to make video while observing it under the microscope, and that too in high power. Here these are the structure. It's some still photograph. आपको यहाँ पे दिखाई देगा. This is the sealed cell. Antheridial filaments are clearly visible. Here antheridial filaments are clearly visible. Here antheridial filaments are clearly visible. In the next slides, what we have done, we try to press it. हमने जैसे press किया इसको so that this will get breakdown. Sealed cell breakdown हो जाए और ये बाहर आ जाए. So are sealed cell are visible here or not?
Yes, sir. It's transparent.
Visible तो है लेकिन clearly visible नहीं है. These are the released antheridial filaments, and each of these cells will give rise to single anthrozoids, biflagellated anthrozoids. This is the permanent slide. They have used some other stain. इसको भी देख लीजिए आप एक बार. Then you will discuss the rest of the part. यहाँ पे और क्लियर होगा क्योंकि ये परमानेंट स्लाइड्स हैं, टेम्परेरी स्लाइड्स नहीं, बेटर मटेरियल है. Everything is quite visible. Secondary laterals, globule, nucleus, antheridial filament, egg cell, corona, nodes, internode, cortical cells, everything is visible. Okay. Never fix your slide in this direction. जिस डायरेक्शन में हमने ये अभी दिखाई है आपको, never try to fix your slide. It should be upside down, not downside up. This is the bottom side, but here you can see the how beautifully they are arranged. Try to freeze it over here so that you can see how these filaments are attached with the nodes or with the manubarium. यहाँ पे आपको दिख रहा होगा मैन्यूबिरियम बीच में. इस पार्ट को थोड़ा सा बड़ा करके देखिए. You can see this is the attachment of this antheridial filament with the node or with of limited growth. आपको आंसर पता लगा कि why we have used two different stain, iodine and acetocarmine?
Yes, sir.
Why we have used two stain? Because when we want to see the storage material, where we will find the storage material? In the egg cell, क्योंकि उसी को नरिशमेंट देना है, उसी को पाल पोस के बड़ा करना है, जर्मिनेटिंग जाइगोट को, डोरमेंट जाइगोट को. So there we will see huge amount of starch. That's why it is taking more stain of iodine. Whereas the antheridial filament, it don't have to perform too much of task, so that will not take more stain of iodine. But the antheridial filament are having mainly the chromosomal part. That's why we will stain it with the acetocarmine. जब हमें क्रोमोसोम देखने होते हैं, जब आप माइटोसिस मियोसिस करोगे ना, then we will use acetocarmine. Because here our main function to observe those structures which are having more chromosomal part compared to any other cell structure.
Yes,
So
sir.
this is the beautiful diagrammatic representation. I have taken this photograph from the HD Kumar book. So now you can see this is the corona, one cell, second cell, third cell, same the fourth cell picture. Now you can imagine how they are twistly arranged. So this is the diagrammatic view of globule and nucleus on a main axis. An enlarged view of globule. This structure, this is a simple bifurcated spiral sperm. Having the pedicel which give rise to the manubarium, from which it gives rise to primary capitulum cells, and from those primary capitulum cells it will give rise to the these filament, and from these antheridial filament each cell will develop the anthrozoids, and anthrozoids will be biflagellate. Fertilization, when it's the time of fertilization, they will separate out from each other. They will get separate out from each other, and then this male sperm will move toward this egg cell, and from the area जब ये लूज हो जाएंगे तो both the tube cells will give a space so that the male sperm or the male anthrozoids or the male biflagellated spiral sperm can comes in the contact with the egg cell, and once they will get fused with each other, they will not allow the entry of any other spiral cell. So that we have discussed here. In a mature nucleus, at the time of fertilization, the 5 tube cells separate from one another just below the coronary cells, resulting into the formation of 5 narrow slits. And several anthrozoids may enter the nucleus through these slits. One anthrozoid penetrates through the gelatinous ogonial wall near the receptive spot. किसी एक जगह जहां तक वो एंट्री करेगा वहां तो फर्टिलाइज कर देगा, but the moment it gets fertilized, the rest of the anthrozoid will not be able to fertilize the same egg cell. Now the zygote will become an oospore after secreting a thick wall. oospore मैंने आपको बताया जब भी हम ऊ की बात करते हैं means बड़ा, it is a big zygote with having a thick cell wall, and now this zygote will known as oospore, and it will survive during the unfavorable condition. Okay, and where it will go? It will be on the bottom of the pond, and it's germinate when the environmental condition is suitable for the germination. The zygote nucleus migrate toward the apical portion, and it will divide twice. Meiosis होगा इसके अंदर. That's why twice. First is the reductional division, forming 4 haploid daughter nuclei. One nuclei will be one side and 3 nuclei will be other side. And oospore is the only. The oogonian alveolar burst open at the apex, and the lenticular cells get exposed. So this part will get known as the lenticular cells, and these cells will divide and give rise to the rhizoidal initial and the proteinumin initial. एक तरह से इनका जर्मिनेशन होता है. ऑल्दो इतनी डिटेल जानने की जरूरत नहीं है, आपको ये पता होना चाहिए फर्टिलाइजेशन कैसा होता है. At the time of fertilization, corona cells or the tube cells will give space to the male antheridial cells, and when it will get fertilized, it will not allow the other sperm to get fertilized with the same egg cell, and now it will known as the oospore. This zygote known as oospore because of its thick cell wall, It will remain in the bottom of the pond and survive during the unfavorable condition, and when the condition is favorable, it will germinate, and it will germinate after the meiosis, and the meiosis, it will give rise to two initials. One is the rhizoidal initial, which give rise to the rhizoidal part or the underground part, and the protonymal initial, which will give the above ground. You will never say aerial part. कंफ्यूजन हो जाता है, कई बार बच्चे बोलते हैं एरियल, एरियल में नहीं होगा, अभी अंडर वाटर ही होगा, बट वन इज इनसाइड मड, वन इज अबव मड. This is the life cycle we have already discussed. So the entire life cycle we have seen that zygote or the oospore is the only diploid structure. That's why its life cycle is of which type?
Haplontic type.
अभी तक जितनी भी पढ़ी है हमने सब में हैप्लॉन्टिक लाइफ साइकिल थी, बिकॉज दिस इज द मेन फीचर ऑफ ग्रीन एल्गी. जैसे डायग्रामेटिक व्यू हमें पता है वेजिटेटिव रिप्रोडक्शन होता है, सेकेंडरी प्रोटोनिमा, एमोर्फस bulbils और एमाइलम स्टार, and sexual reproduction will take place by globule and nucleus, which both are haploid. One will give rise to anthrozoids, another will give rise to egg cell. It will get fertilized, will form zygote or oospore. This is the only diploid structure, and it will undergo for meiosis. Okay, and meiosis will give rise to protonema, and okay, so this protonymal initial will give rise to the chara, and the rhizoidal initial will give rise to the rhizoids of this chara. So this is all about the life cycle.
सर कोरोना सेल्स एक बार बता दीजिए.
कोरोना सेल्स चलिए. Inside this is unicellular egg cell or the haploid egg cell. इसके जो चारों तरफ जो ट्यूब लाइक स्ट्रक्चर है, so this is the corona, the upper terminal part of these tube cells, and these tube cells will twist, wrap this egg cell in a twisted form. तो 1 2 3 ये इसके पीछे वाले सेल 4 5, ये 5 सेल ने ट्विस्ट लिया, फिर घूम के फिर 5 सेल आ गए, फिर 5 सेल्स आ गई. ये इसका मैनर होता है पूरा ट्विस्टिंग.
यस सर.
आप इससे इमेजिन भी कर सकते हो कोरोना के ऊपर जो आउटग्रोथ होती हैं, दे आर आल्सो गिव्स द सेम काइंड ऑफ अपीयरेंस. दैट्स व्हाई इट इज नोन एज द कोरोना वायरस. लाइफ साइकिल तक हम आ गए थे. क्लासिफिकेशन पैटर्पोइड कैरोफाइसी तक सेम है, ठीक है, जो हमने कोलियोकीट के लिए किया था. एंड कैरेल्स के लिए हमें पता है कि बिकॉज द कैरेल्स इज द ओनली ऑर्डर एंड द कैरेल्स इज द ओनली फैमिली, sorry, Caris is the only family of this order Careals, and that's why we are justifying both the things, order as well as family together. And how we can justify it? That Cara is a member of Careals or Caraceae, no asexual reproduction, we haven't seen that. Oogamous sexual reproduction, sterile cells around Antheridia and Oogonia, we have seen that. आपने देखा होगा कि चाहे वो egg cell है उसके ऊपर भी spirally twisted tube cells होती हैं, या फिर जैकेट्स बनाती हैं एंथीडिया की, और जो कवर बनाती हैं. मेल गैमेट्स कवर्ड विद द स्केल्स, जूस्पोर नोट प्रोड्यूस, वी हैवंट सीन दैट. कॉम्प्लेक्स प्लांट बॉडी विद एपिकल ग्रोथ एंड डिफरेंशिएशन इनटू नोड्स एंड इंटरनोड्स. So these are those features which makes Cara as a member of this Careals or Caraceae. So you can note down this justification from here. ये एक स्लाइड बनाई है. You can have a look at how the male and female reproductive structure, that is globule and nucleus, originate from the initial cells. Here it is the better representation. Means this is the antheridial filament. These are the antheridial cells, and inside this antheridial cell you will see that anthrozoids or sperm.
सर ये हम लोग कोलियो हैं सर.
हाँ, ये कोलियोकीट में लिख चुके हो ना, तो either write
यस सर.
group eukaryotic algae, phylum chlorophyta, class Carophyceae, and then just write a single line as per coleochaete. If you have already written it and you don't want to write it again, simply write as per coleochaete classification. Our main purpose of writing this justification is to show you how one algae is different from other. अगर हम ये नहीं बता रहे हैं कि क्लोरोफिल A and B है, that means we don't know it. If we are not mentioning that whether it's dividing in node and internodes, it is having highly एडवांस प्रोगेमस टाइप ऑफ सेक्सुअल रिप्रोडक्शन, जिस्पोनोड प्रोड्यूस। ये स्लाइड्स जो आपके होती है जस्टिफिकेशन वाली, समरी ऑफ द एंटायर थिंग, आपकी पूरी समरी है ये उस पर्टिकुलर जीनस में जाओगे। इफ यू बिकम ए फाइकोलॉजिस्ट एंड यू कलेक्ट एल्गी, देन यू विल use the same pattern. Okay, it's a eukaryotic algae or prokaryotic algae? Okay, it's a eukaryotic algae because chloroplast surrounded by the two membrane of the chloroplast envelope. Chloroplast इसका मतलब हमारा eukaryotic है। उसके बाद हमारा है क्लोरोफाइटा। Why it is क्लोरोफाइटा? Because it is having both chlorophyll A and B. स्टार्च भी इसके अंदर है। Okay. Why it is carophysi? Because इसके जो reproductive structure they having the laterally placed subapical inserted flagella. सिमेट्री नहीं है उसके मोटाइल सेल्स के अंदर। राइजोप्लास्ट इज नॉट देयर। इफ व्हाटएवर फीचर्स यू कैन सी, इफ यू विल ऑब्जर्व इट अंडर द लाइट माइक्रोस्कोप, तो आपको राइजोप्लास्ट से आई स्पॉट नहीं दिखाई देने वाला। ओके, आपको फ्लैजिला नहीं दिखाई देने वाला। बट व्हाटएवर यू आर ऑब्जर्विंग बेस्ड ऑन दोज फीचर, कैन यू डिवाइड इट? इफ यू कैन डू, जस्ट गो फॉर दैट। इफ यू कांट डू, देन ट्राई टू गेट मोर इंफॉर्मेशन। सब तरीकों से पता करते हुए देन यू विल प्लेस योर एल्गी इन ए पर्टिकुलर टैक्सोन, कि किस ग्रुप में, किस फाइलम के, किस क्लास के, किस ऑर्डर के, किस फैमिली में आएगी। एंड फाइनली वंस यू रीच अप टू द फैमिली लेवल, तब आप लिटरेचर सर्च करना स्टार्ट करोगे कि मेरी एल्गी बाकी के जीनस से अलग है या नहीं है, कि मैंने कुछ नया तो नहीं खोज लिया। आज आप एक एल्गी लेके आओ, आपको कैसे पता कि इट्स ए यूनिक एल्गी? आज से पहले आपने देखी है किसने या नहीं देखी है? तो फर्स्ट यू विल narrow down it. You can use any classification. You can use Rich classification, you can use Smith classification, you can use Lee classification or any other classification. Okay, but every classification will tell you how you will place it in a particular group or family. The first diagram you will make the morphology. This diagram.
Yes, sir.
Yes, sir.
The second diagram you will make the branch of limited growth. This one, only this one. Third diagram you will make this diagram. So these three diagrams will be there. So that's it from our side. But for rest of the diagrams, they will be there for your theory purpose. हम तीन diagrams बता रहे हैं only for your practical record, for your practical record. Whatever you can observe it under the microscope or by your naked eye. आपको ये नहीं दिख रहा है primary protonema और primary rhizoids. It doesn't mean that you will not remember this. You have to remember this, especially for your theory purpose. In the same way for life cycle, at least diagrammatic representation of a life cycle you should know. So that's it from our side for today.